How's it going, y'all? Jack Mycologist here, back with another video. Today we're going to be talking about mushroom breeding and reproduction, specifically for Basidiomycota, right? And cubes are part of that, so we're going to be talking about it. Hopefully this explains everything relatively simply and gets the points across that I need to show you. And yeah, so if you just want to learn about this process, this is a good starter video, I feel like. So let's say you have a multi-spore source, whether that be a syringe or a swab or a spore print. These are covered / filled with tons of spores, tons and tons and tons. You see all these little dots representing spores here. Okay, let's say you grab a single one of those spores, single one, bring it up here and you zoom in. You'll see this little dot represents a nucleus. Let me move this over here. The single dot represents a nucleus, so it's one nucleus within this spore. That is a monokaryon. Mono meaning one, karyon meaning nuclei. Okay, let's just remember this part extra today. Karyon, nuclei. Kary, nucleus. So it's just karyon is the plural, kary is singular. So let's just highlight that again. Karyon, nuclei. Kary, nucleus. Okay. So continuing, let's say you put a spore in the right environment with water and or nutrients, it'll undergo germination and closed mitosis. Plants and animals do open mitosis, which is has to do with the nuclear envelope, but I'm not going to go too much into that. You can go Google or YouTube a video for that. So it undergoes mitosis and begins making these chains of identical cells. You zoom in and it'll look like a tube. These tubes are called hyphae. The stringy tubes are called hyphae. They're separated similar like bamboo, and those walls are called septum. Okay, and throughout this hyphae, it is genetically the exact same. Each cell is daughter cells, and they are the exact same genetically. So we're going to call that monokaryotic hyphae. Monokaryon, karyon meaning nuclei, one nuclei, hyphae. Again, monokaryon, one nuclei. Pretty simple. So we're going to go over here real quick. So let's say we're going to treat one egg as a spore or one sperm as a spore. Okay. Fungi don't have sexes. They can have anywhere from 3 to 20, in fact. So they just use matters of plus and minus to represent genetically different hyphae. So fun fact also, unlike most fungi, Basidiomycota reproduce sexually. So it takes a plus and a minus connecting to do all the rest of this process, which I'll explain. So let's say you have an egg which contains 23 chromosomes, and you try to combine it with a genetically exact same egg with also 23 chromosomes. It will not have enough new combinations to make the 46 chromosomes needed for a person. Not equal. Okay. Same thing goes for a sperm. Twenty-three chromosomes does not equal a human with 46. Okay. So same thing goes for if we have a, let me erase all this real quick. Sorry, if just go back and take the screenshot if you need to. So if we have make this one bigger. Plus, I don't want to make this too confusing. A plus culture plus another plus culture does not equal enough genetic material to make a fungi. Same goes for minus, plus, minus. There we go, sure, something like that. There we go. Does not equal enough genetic material to make a fruiting body and reproductive cells. So as we come back, we'll have two genetically different hyphae here. You can see by the colors. We'll have plus and minus, okay? They're going to undergo syngamy as they combine, which is this whole process here, okay? First is plasmogamy, which is when the two cytoplasms of each hyphae form together and bring the nuclei closer. And then karyogamy, which is when the two nuclei combine and form one nucleus that is genetically different than the parents because it's a combination of both. Okay, and we're going to call this 2N from now on. 2N, each one of these little cells here. 2N means two nucleus. So you could say the previous monokaryon is equivalent to 1N. The reason this all happens is as they come together, they'll form these little bodies called clamps where they'll use it. Let me grab my other color here real quick. Right, so that's part of the whole process. Is zoomed in, zoom in of these hyphae here with the clamps, and these are what's referred to, as clamps. So when people say they're looking for clamps under a microscope, that means they're looking for these little bumps on the side of the hyphae that indicate that syngamy has taken place. So as the hyphae condenses and grows, it will begin to form mycelium, okay, which is just the weave of hyphae, okay. And we're going to call that dikaryotic mycelium instead of 2N mycelium, okay? They mean the same thing, but we're just going to call it dikaryotic mycelium, okay? And this has enough genetic information from these two parent cells that it can now form reproductive cells and a fruiting body, okay? And that's what we know as mushrooms. So as the fruiting body continues to grow, it'll form gill tissue, which is condensed mycelium. This mycelium will then form the reproductive cells called basidium, right? In this basidium, they look like little clubs with forks at the end, and the spores will form at the end of the forks. This basidium will force meiosis to happen, right? If you want to learn about meiosis, go check out Amoeba Sisters. They have a video about it, and it's quite helpful, shows all the visuals, and explains the whole process fairly well. So they will undergo meiosis and form 4 new daughter cells that are spores. Each one of these is a unique combination from the original 2 parents, but each is genetically different from each other, right? And these are 1N now. They are no longer 2N. They've been split up, made combinations, and now 1N. And 1N, again, means one nucleus. One nucleus, I've heard that before. Back at the top, monokaryon, 1 nuclei. So 1N will start the whole process that we just did over again. Hence the repeat process, okay? We're going to start this whole process over again from this 1N spore, okay? So it'll go across the substrate, wherever it's on. The spores will spread across, form their little hyphae together, and they touch, dikaryotize, and form, again, dikaryotic mycelium, which then has the genetic potential to do all of this over again. And it continues in a cycle. So yeah, hopefully that explains this relatively simply while showing all the important things. If you guys have any questions, let me know in the comments. Feel free to hit me up on Discord as well if you have any questions about it. I'd be happy to explain anything that I know, or be educated if I said something that was a little off. So just let me know. And I hope you guys enjoyed this video. Hopefully we all learned a bunch, and I will see you next time. Peace.
Mushrooms rising under moonlit skies, Dikaryotes bloom where the mystery lies. In a monotone we build a sanctuary.